Okay, our next talk, we're going to switch kind of to lower trophic levels. Dr. Russell Hopcroft is our next speaker. He's a professor of oceanography at the University of Alaska Fairbanks. Russ earned both his master's and his PhD at the University of Guelph in Canada. And then he moved on to a postdoc at the Monterey Bay Aquarium Research Institute. And from what I understand, that's where he started to get quite involved in both deep sea work as well as remote sensing and ROVs. So Russ has a strong interest in zooplankton, as he states, from common copepods to gelatinous forms that are very difficult to see and to characterize through standard techniques. And so he's spent throughout his career quite a bit of time looking at developing and implementing or using remotely operated vehicles and doing in situ and video technologies. And that's what he's going to be showing us today. He's definitely well known for images and videos of live zooplankton. If you pick your favorite taxa or species of zooplankton and Google it and look under images, chances are you're going to find one of Russ's photos as one of the first hits. They're also really beautiful. Here's an example, and Russ will show us quite a bit more. So the title of his talk is A Deep Dive into the Zooplankton of the Gulf of Alaska. So Russ is going to take us out to seamounts about 200 miles or 200 miles offshore, and then looking at a variety of taxa within the water column from the surface down to the abyssal plain about 4 kilometers down. He's going to be outlining some of the tools that they use, some of the analyses that they apply to process that data, and then also providing some images and videos of what they see down there. In this talk, I believe it's mostly more on macrofauna, so jellies and ctenophores and squids, including some new species to science. So Russ, I'll let you take it from here.
Okay, it's nice to see you all here with the weather being like it is outside today. I know it was a challenge for somebody to get here. As Kathy said, when we stand up here, give these presentations, we're really representing a whole bunch of people, and we're just the spokesman. I in particular want to thank the staff in my lab, Caitlin Smoot. I want to thank Jennifer Questel and Dougal Lindsay from Japan, who are kind of culprits in this seamount activity, and all the plankton that shared or gave their lives in the process of this research. The work I'm going to be talking about today is a combination of different funding sources, NPRB, NSF, the LTR, and in particular, NOAA's Ocean Exploration Program. So zooplankton come in all shapes and sizes, ranging from the crunchy crustaceans that we have over on the left to semi-crunchy things to things that are very soft-bodied and difficult to study or difficult to identify and work with. There's a lot of challenges that people don't appreciate in working with zooplankton. We have an incredibly large size range. We think on land of a mouse to elephant being a big difference in size. That's about 100 times body length. The difference between a copepod and some of the larger jellyfish is 10,000 times. The large range and fragility. Crustaceans would probably be best described as men in armor, and jellyfish would be described as jelly. And you can imagine that very different tools will collect both of those in good shape. There's a large range in mobility and escapability. There are some things that are really hard to catch, and there's other things where you'd have to be inept not to catch them. And there's a huge range in abundance. Copepods we measure in thousands per cubic meter. Kingston Harbor, Jamaica has 250,000 copepods per cubic meter. And large jellyfish may be 1 per 1,000 cubic meters. That's a million-fold difference in abundance, which means it's really hard to describe the full range of a community. So how do we study zooplankton? The way we always think about is kind of traditional plankton nets. It's based on morphology. It's the way we've been doing things for hundreds of years. Starting in the 60s and 70s, we started kind of getting into the sexy science. We started using scuba. We started using ROVs and submersibles. More recently, there's a big push on using molecular tools to identify plankton. And there's also, in parallel, in the last couple of decades, been this push to in situ imaging of zooplankton. So these are our suite of tools that we can use. Each has its inherent biases. No one of these is perfect. Every one of it has its shortcomings and its strengths. Plankton nets are what I call the catch and kill part of science. It's really easy for us to go out and get them, bring them back to the lab, and that means the heavy work is done off the ship. They're simple. They're basically big butterfly nets. They can be very inexpensive, or you can get the Cadillac version of them that costs a small fortune. They're quantitative, but we always have this trade-off between trying to get the really mobile stuff, which tends to then squeeze the fragile stuff through the net, or getting the fragile stuff and the mobile stuff avoiding our collection. So no single net can assess the whole zooplankton community. And most people will use at least 2 types of nets to try and collect the zooplankton. If they're lucky, we'll have an even wider selection of tools. Damage occurs during collection and preservation. It's not a perfect approach. And if we had to summarize what are nets good for, they're really good for copepods. They're kind of so-so for krill because they like to jump out of the way, and they are not very good for jellyfish. But nonetheless, they remain the workhorse of zooplanktonology. And when I get up here and talk about the Seward line, I'm going to be primarily showing you plankton net data. So eyes in the water. So one of the first ways we started realizing nets don't cut it is we started putting scuba people in. You can make this semi-quantitative. You take your vision, you can go in and you can collect stuff. When we get outside the range of what a diver can do, we put you into submersible or we put you into an ROV, and you basically control where you're looking in the water column. This works really well for large and fragile stuff. Doesn't work very well for the small and the abundant. Can also use molecular sequencing, and this is getting to be a popular approach. It has the potential to identify everything in the sample, regardless of how old it is. Juveniles of almost everything are hard to identify. Bulk sequencing is only as good as the lookup library. So it's like going out collecting license plates in the parking lot, and that alone won't tell you what kind of car they were attached to. Right? The license plate needs to be keyed to the car it goes to. And at present, it's kind of hard to scale molecular data to give you the traditional kind of abundance or biomass that we want to base decisions on plankton community structure on. Tells you who's there, but not so much how much. The big thing that's happening now is in situ imaging. These are examples from our system that we are using in the Gulf of Alaska. It's great for fragile as well as robust species. They've now got some fairly high definition in these. It is quantitative, but we end up with the same trade-offs about how much water you can look at and what kind of magnification, and that will bias what you learn from this technology. Where are the knowledge gaps in marine life in general, but especially for zooplankton? Most of what we know about the oceans is confined to shallow waters over the continental shelf, and it's confined to the areas, as this heat map shows you, of the shallow ocean. It's really not very good in deep water, and it's not very good once you get off the continental shelves. When we do sample that oceanic habitat, it really is limited to the upper couple of 100 meters, because you get unpopular if you take hours and hours to collect a plankton net deep in the water column. So the bias is against the deep, the rare, and the fragile when we use this approach. or what we know at present. So what I want to lead into now that I've given you the backdrop is some work that we did at the Seamounts in 2019. We had the unique opportunity to go out, basically extend the Seward line way offshore to a series of seamounts that are about 200 miles at the very edge of the US EEZ. We sampled 4 stations from the surface all the way to 4 kilometers down in the oceans. And we also looked at the communities on top of the seamounts. And I'm going to be reporting you very much work in progress. We are not done with this yet. So these seamounts rise from the abyssal plains at about 4,000 meters up to flat tops at about 600 to 700 meters. Once upon a time, they were actually at the ocean surface and they're covered in sandy tops. And the real question is, how do these seamounts operate? Are they enhanced productivity zones, or are they different, or are they just consistent with the surrounding habitat? The tools we use the ROV Global Explorer, which is shown here. It's about the size of a small car, and it comes equipped with a high definition camera, which allows us to look very closely at the zooplankton and a zoom lens. And it allows us to collect animals to bring back and confirm what they are after we see them in situ. We also paired that with traditional plankton nets. For example, this mock nest, which has got a 1 square meter mouth, and we tow it, which means that it's getting the fast mobile stuff and not so friendly to the fragiles. And a pair of nets that are pulled vertically more slowly and are more friendly to the fragile stuff, but not ideally perfect. Both of these have got multiple nets, so we can carve the water column up into the different layers. And we can, because they bring samples back, we can look at both traditional morphology and molecular biology to help us learn something about the taxonomy. So once they come back up, we go into the lab, we do live sorting, particularly for the vouchering for molecular identification, and we take lots of pictures, and then we will use that to try and piece together what's going on in the community. Some of the patterns that have come out of the plankton nets are that diversity tends to increase with depth in the oceans. You can see there's often twice as many different species present in the mid part of the water column and less actually at the surface. The species become more different the deeper you go. And there are differences actually between the shelf and the deep sea. There's actually more diversity in the offshore water where the productivity is low compared to the inshore water where the productivity is high. The problem is that once you start working with deep sea material, it's not well known. There aren't good taxonomic keys, and there's a huge learning curve to start getting a good handle on what's actually living there. And we severely underestimated the effort that was going to be required in this project to do a good job with that. And as I said, every technique's biased. So if you collect with nets, what you're going to discover is that copepods rule everything you're going to do. All right. So whether we use the coarse net or the fine net, and the coarse net starts being a little bit more reflective of these rare and big things, but there's this bias there. We do get some surprises in nets. So this is a kind of like a selp. It's called a doliol. It's a pelagic tunicate. We've been seeing something in the nets since I got here about 25 years ago that was so heavily mauled when we collected it in a plankton net, we really couldn't say what it was other than what phyla it was from. And we went out with the ROVs, we were able to collect and fine mesh nets, we were able to collect some of the stuff in good enough shape to ID it. And this is a Doliolula equus, is the most abundant large multicellular organism at 400 meters, 300 to 400 meters in this ecosystem. We didn't know that till we used the right tools. Jen Questel presented some of the results of the molecular stuff last year at AMSS, And we're making a lot of progress in building up that library so that when we do whole sample barcoding, we can get a handle of what's there. And that's one of the essential things that we're working on in this project for the deep sea. We're going back in just 4 months, 5 months from now to start doing some of it over again. We're going to do more plankton nets. We're going to do more DNA barcoding. We're going to add a small fish trawl with a camera in the cot end to see what we're getting and what we've been missing by not having that tool. And we're going to be taking this imaging device, the deep plankton imager shown in the picture, and it images a cubic meter of water every 4 seconds down to 50 micron pixel size. And with that, we're going to get a handle on the really fine scale distribution of everything from copepods up to medium-sized jellyfish in this system. So why do we need so many tools when we study zooplankton? Most of you know this joke. I'm not supposed to tell a joke here, but I can allude to one. So if we put a whole bunch of scientists and blindfold them and we send them in the direction of an elephant, each of them is going to describe the part of the elephant they see differently. So we need each of these tools to describe different parts of the elephant and then synthesize that into what's really going on with the zooplankton community. So each of these tools has its biases. So the DPI can get the small and the fragile, but it can't get the big and the rare. The DPI also just collects images. We don't get anything for DNA barcoding. We don't get anything to be absolutely certain what it was, even if we get a good spatial distribution. The ROV is biased toward just the big stuff and the fragile stuff. The plankton net itself is biased toward the small and the abundant and the robust. And a fisheries trawl, of course, sees a completely different cross-section of the community. Surprise, it emphasizes the fish. So with that, let's try and look at some video, and we're going to dim the room a little bit more because the contrast to this works a bit better. Okay, so we're going to start out with the ROV. What we normally see, I'm going to pull this overlay off it so we know the whole dive, what we're seeing for depth, temperature, salinity, headings, and all this other information. And what we start to discover is a selection of small jellyfish in this system. It's not showing up well. Okay, so this is a small narcomedusae. It actually is a jellyfish that eats other jellyfish. It's trying to swim into them with its tentacles pointed forward. These are super abundant between about 300 and 500 meters in the water column, and they disappear. This species, that species is easily confused with another. This one's a little copepod eater called Arctopodema. The problem with these is they're almost too small for the ROV. They're about the size of a fingernail. So while I can show you they're there, I have real trouble getting a handle on what these small jellyfish are doing in the water column. And this is where we get back up from the plankton nets. Where it gets interesting is when we start getting into the bigger jellyfish. This is Solmissus. It's about the size of a large chocolate chip cookie. Katrin, I got chocolate worked into the presentation. All right, it's a narcomedusae as well. It hunts both in either a swim in with the tentacles or a wait for things to blunder at mode. It eats other jellyfish. And when we get these, and here's where it is in the water column. It's mostly seen between about 300 and 500 meters, but goes significantly deeper. When we get these in the lab, they are a piece of jelly with nothing identifiable besides that on them. But when you see them in situ, you realize how fragile and how delicate and how beautiful they are. Here we can see something, all this tissue that's normally eroded off. Well, it's tumbled around in a plankton net. This is Paraphila. It is a cornate. It also tends to hunt by swimming into stuff. The tentacles are held forward. It's pretty common in midwaters throughout the world's oceans. It's also, we're going to see in the middle when we look up the backside, many this group of jellyfish actually carries embryos of its young. It doesn't just, there they are. Those little white dots you see in the bottom are juvenile jellyfish that are being incubated by the mother. This is confined to around 500 meters. This is Paralia. It's about the size of a dinner plate. You can see from the long tentacles around the outside, it's probably hunting things like small crustaceans. Okay, really narrow size distribution, or sorry, depth distribution on this. This is Pandea. We've only seen a couple of these during the cruises so far, but you can kind of get the impression when we come up on this, it's got tentacles that are going out very, very far. It's about, oh, maybe 5 or 10 centimeters in size. These tentacles are probably going out a couple of meters in all directions. And now that it's aware that we're there, you can see it starting to reel these tentacles in. And it's really interesting to think of this. So think of this as kind of like somebody setting out a whole bunch of fishing lines. This will be feeding on most likely small crustaceans or large crustaceans. So its mode will be to go someplace, spread the tentacles out, fish for a while, pull them back in, swim someplace, and go to a new territory. Of the things we've looked at, this is one of the rarer jellyfish in the Gulf of Alaska. The 2 animals that I've annotated so far are in a very narrow size range. This is a gina. It's very closely related to the Solmunda gina we started out with, much bigger. It's about the size of a big toe in terms of its body size. The orange thing you see on the side is actually a jellyfish an amphipod or a sand flea that is a commensal and tends to like look for leftover food on the outside of the jellyfish. Because jellyfish are so badly damaged during collection, Agina and Solmundagina have been confused when plankton net collections are looked at for decades. Very different in size. When you get them intact, you can see a lot of differences in them. But when they're reasonably mutilated by a plankton net, it's hard to tell them apart. Agina is broadly distributed through the water column. Another Narcomedusae, Agenura, was also found pretty commonly during the dives. This is probably about the size of a teacup down to about maybe just 4 or 5 centimeters. Broadly distributed through the water column. Ctenophores are a little bit underrepresented in this. This is a Baroe. All these things you see that look like light shows down the side are fused cilia that are used as paddles to propel the animal through the water column. Baroe eats other ctenophores. It doesn't eat the hard zooplankton we normally think of. It's eating something like this, is Euplocamus, which is actually out there with those tentacles and feeding on small copepods and small crustaceans. We really don't have good enough data on these yet to be able to describe the distribution that goes with any of these taxa. This is Lampoctinus, a lobate ctenophore. This one actually has almost like a series of catcher mitts, and it uses ciliar action to corral small crustaceans, primarily copepods, toward its mouth. Like all ctenophores, it has 8 rows of these teens or combs along the outside that it uses for propulsion. And I just want to emphasize how delicate the structure is on some of these things to make the point of how else could a plankton net collect them except for in horrible condition. What you're looking at in the middle is its stomach. Some of them are really hard to work with. This ctenophore here, another Lampoctina species, is really, really dark, but when you bring it up and you put it in the sunlight, this is what it looks like. Coming back to the jellyfish, this is a tola. A tola feeds on small crustaceans. It's interesting in that it always seems to trail one of its tentacles behind as kind of its main fishing line. And like the paraphila I showed earlier, it tends to brood young around the bill margin. Seems to have a narrow size distribution, but at times we find it fairly shallow in the water column. Some animals, again, we've only seen a few times during the dives. This is Tomopterus. It's a polychaete worm. This specimen was 1 meter long. I'm partial to larvaceans, which are another pelagic tunicate. The little white thing you see is the actual animal. It secreted this filtering mesh around the outside of it. These are incredibly hard to work with in the ROVs because it's kind of like the bull in the China shop. You really need a gentle touch to go in and start looking at these animals. Don't have a very good idea how abundant they are. Don't have a very good idea of the species composition because they collect in horrible shape with the plankton net. With experience, though, we can tell them apart by looking at their house structure, or if we're really lucky, closely zooming in. Here's an example of a related species, Fritillaria. Neither of these species have been documented in the Pacific since I found them when I was at Ambari. Right, back to the jellyfish, Botronema. This is, if you look at the tentacle structure on it, this is again another small crustacean feeder. An interesting story on this is the species were told apart by the presence or not of this little knob on the top of it. And we discovered that the knob doesn't tell the species apart with molecular work. In fact, the species with the knob we have here in the Gulf of Alaska and the one with the knob in the Arctic, different species. In the Arctic, they thought there was 2 species, one with and without a knob, and they're both the same species. Broadly distributed, same family, Halocreus, pulled apart by having more canals on the body, fairly deeply located. Back to another one of the jellyfish. This is back to being a small crustacean eater. Tychogenia is a little bit deeper in the water column. Okay, this is the size of a volleyball or a small beach ball. This is Paraphilopsis. When you see something like this deep in the water column, you are suitably impressed. We really don't have a very good idea of what this is eating to sustain its size. Everybody goes silent when you come across these things with the ROV too. But despite its size, it's surprisingly fragile and easy to damage. So far, we're finding them reasonably deep in the water column. Another one the size of a beach ball. This is Tiburonia. Tiburonia was actually only described about 20 years ago from the work at Ambari with ROVs. Kind of get a look at we could call it maybe the Death Star jellyfish. Slowly trying to pan around on this. And you can see those little waves that are going through it are just being hit by a little bit of turbulence off the ROV thrusters, which again makes the point this is not robust. If we get this in a plankton net, it will probably be unidentifiable. And now we're kind of getting to see the other end of it. Tiburonia is also fairly deep in its distribution and surprisingly abundant here in the Gulf of Alaska. This is Apollinaria. It's a colonial jellyfish. It's made up of a lot of individuals. I showed it with the tentacles out. Here it is with the tentacles all retracted. What you're seeing are a whole bunch of stomachs on the animal right now. This is actually a combination of two species, which makes it look broadly distributed. Some of them are pretty mobile. Each one of the individuals at the front of this is a little jet propulsion unit. You can see us struggling to try and catch it with the ROV. This is Maris. This is another example of a colonial jellyfish. They are actually aware of the fact we're there. You can see it throwing parts away, trying to confuse us as we come in to take a look at it. Again, it's got these tentacles and mouths in the back half and these swimming bells at the front. You can see it getting really upset and falling apart as we look at it. And again, this species is pretty deeply distributed. Okay, you can never go wrong by showing a cephalopod. Okay, this is known as a cockatoo squid. They're kind of like deer in the headlight shot here right now. Normally they're out kind of at the edge of the vision and we're trying to like, they're out of there. And every once in a while they go, and then we get to zoom in on them and do some nice video. They're very charismatic. And we probably blinded that animal for life. Okay, we're getting a nice look at them right here. The cockatoo comes from the tentacles are all held together and give you that kind of cockatoo feather impression at the back. I got a colleague who used to work at the Smithsonian. His comment was you could never do badly by ending your video with the cockatoo squid. I think you can't do badly by ending it with the Solmissus. And to sweeten the pot, these fireworks are us putting it through a laser sheet to try and get an idea of some of the structure and the size. But for those who disagree and really think you need the end with the cephalopod, I figured I'd put the cephalopod back in again and let you think about how cool some of this stuff is that you can find deep in the ocean. And that's what I've got.
Thank you, Russ. Do we have some questions from the audience?
I can't see anything, so.
Yes.
I didn't hear the question.
What eats these deep sea predators? And if you just repeat the question.
What feeds them?
What eats them?
What eats them? Ooh. Well, a lot of the jellyfish I showed you are eaten by some of the other jellyfish I showed you. So that's kind of the easy part of the answer. There are fish that are specialized on them, something like that cockatoo. We think it's more likely eating small fish. There are probably some big filter feeders in the ocean in the way of things that they're capitalizing on them. But it's always surprising to hear that most jellyfish are eaten by other jellyfish. Things like tuna are actually surprisingly capable of feeding on a lot of the gelatinous zooplankton as well, but they don't tend to hunt so deep.
Other questions?
Did you take that video in a single dive? Or is that
No, this is a compilation. Most of the dive looks like where I started out, where you see all these specks coming by you of nothing for like minutes at a time. And but those specks are all the things I normally talk about. They're the copepods and the larvaceans and the other things that at this magnification, they look like they're nothing. So this is, I harvested this out of 3 dives. So about 18 hours to 24 hours of video distilled down to some of the best stuff that was in focus. This makes it look like it's actually much simpler than what it really is. Best way to describe it, imagine me trying to tell somebody to take something the size of a small car and have enough control over it to let me look at something about the size of a marble. And so it's really hard to get stuff in focus, to get it framed properly. and to end up with it before it gets dusted by the thrusters, creating backwash. Kathy?
Are you able to use these videos to get an indication of abundance? Do you use that?
Yeah, the approach with the ROV is semi-quantitative, so it allows us to establish the depth range and kind of commonness of something within it. There's another step required where we have to. So if we take out the time where we're not looking for something, i.e. we're zoomed in on something, taking pictures of it, and where we're focused in too tight, we can reduce it the amount of time in kind of search mode, and then take time at depth and standardize the catch per unit effort to time at depth. And that makes it kind of semi-quantitative, but that's one more layer into the analysis and what we're at right now.
I'm curious for some of the more delicate species that you're able to capture. Are you successful in transferring those to aquaria for future study or preserving them in some way?
They don't live very long, the fragile stuff, once you get it back up. I mean, Ambari's tried for years and they've gotten good at some of them, like their jellyfish exhibits. But we learn a lot about them when we do get them intact. We study them and take as many pictures and notes as we can to document the anatomy. Some of the stuff doesn't preserve at all. The moment it dies, it disintegrates regardless of whether it's in a preservative or not. So video records are really the only way we can describe the species and keep a record of it. Yep.
Show that some of these species are pretty limited in their depth range. What are some factors that limit the range of the
We're not. Some of them are pretty limited in their depth range. Do we basically know why? At this point, not really. There's changes in some of the particle densities, and if some of these are predator-prey relationships. So I think that's why you're finding some bigger species in some of the zones. We saw smaller species. There's also zonation in the small stuff that I didn't show you, the typical kind of stuff we'd assess with plankton nets. So that's the ultimate analysis when we finish collecting all the data and getting it ready from each of the different tools, is to then go look for the cross correlations. So. Yep.
Thank you, Russ. Can you briefly compare the seamounts with the more regular abyssal plains?
The problem we had trying to compare the seamounts was the surrounding, was that when we use plankton net technology and ROVs, we only end up with a couple of points at which to compare. So the idea with the DPI is it's a towed undulating system. So we can actually create a big section through the water column based on many profiles and then look for the footprint of the seamounts. So one of the reasons we're changing from ROVs to the DPI this year is to try and see if we can see that footprint in the physics, the chemistry, and the biology that we just didn't feel we could have any confidence on with the ROV. Yep.
I was wondering if you're measuring in any way whether all these little critters are moving around as.
Can you use the microphone, please?
Are you measuring whether these critters are moving to different places as ocean temperatures rise and fall?
We know so few about these things. We couldn't even tackle that problem. There are a handful of places in the world that people have really well described the deep water communities. The ultimate one is Monterey Bay, California, because that's what it does full time. Well, one of the many things it does full time. There's a place off Japan. Woods Hole's got a big effort going on right now for looking at the mesopelagic realm. And the Gulf of Mexico knows a fair bit about what's going on deep, because they had this thing called an oil spill back there a few years ago that forced them to look at those communities really thoroughly for the purpose of assessing damage and trying to figure out what was potentially impacted by that blowout. So we don't know. It's such an unknown realm.